back to our webinar series. Today we are going to talk about flow cytometry, FC, which is one of the techniques that we have at our facility, the BioLab. The BioLab is located at the Nova School of Sciences and Technology and gathers in a single unit a unique set of state-of-the-art biophysical and biological techniques, namely circular dichroism, differential scanning calorimetry, flow cytometry, microscale thermophoresis, and multiparametric surface plasmon resonance. If you are interested in knowing more about us, our website is in the description box of this video. In this presentation, I will talk about the principles of flow cytometry, the major components of a flow cytometer. I will define some common FC terms, present some fluorochromes, discuss fluorescence compensation, and data analysis. I will also present to you the instrument that we have in our facility and give some application examples. Flow cytometry is a technique that measures and then analyzes multiple physical and chemical characteristics of single particles as they flow in a fluid stream through a beam of light. FC has the ability to measure the optical and fluorescence characteristics of a single cell or any other particle such as microorganisms, nuclei, and chromosome preparations. Size, granularity, and fluorescent features of the cells derived from either antibodies or dyes are also examples of parameters used to analyze and differentiate the cells. Traditional flow cytometers are made up of three subsystems. The fluidic system, which transports the sample to the flow cell and eventually discards as waste. The optic system, that includes lasers, mirrors, and lens, and photomultiplier tubes to detect the emitted fluorescent light and scattered light, and the electronic systems, that converts the data into digital signals that can be visualized in a software program for data acquisition and analysis. Now, I will define some common FC terms starting by light scattering. Light scattering is a term referring to physical process involving the interaction of light and matter. When a cell or particle passes through a focused laser beam, it refracts or scatters light in all directions. Light scattering is directly related to structural and morphological properties of the cell. The forward scatter or low-angle light scatter is the light that is scattered in the forward direction as a laser light strikes the cell. The magnitude of forward scatter is roughly proportional to the size of the cell or particle. Indeed, this data can be used to quantify particle size. Side scattered is defined as the light that is scattered at larger angles. Side scatter is indicative of the granularity and structural complexity inside the cell or particle. Other important concept is fluorescence. As you know, fluorescence is the emission of light that occurs when an emitting particle, such as a fluorophore-labeled antibody, absorbs light from another source such as the intercepting laser beam. When the particle absorbs the intercepting light, it is elevated to an excited electronic state, and as it returns to its ground state, the absorbed energy is radiated, where most of it is emitted as light. In flow cytometry, cell populations can sometimes be separated based on forward scattered and side scattered, but cells can also be separated by whether they express a specific protein. In this case, a fluorophore is usually used to stain the protein of interest. In flow cytometry, laser light is usually used to excite the fluorochromes. These lasers produce light in the UV and/or visible range. Fluorochromes are selected based on their abilities to fluoresce with the wavelengths of light produced by the lasers. There is a wide variety of fluorescent reagents utilized in flow cytometry. These include fluorescently conjugated antibodies, quantidots, DNA binding dyes, viability dyes, ion indicator dyes, and fluorescent expression proteins. Fluorophores emit light over a range of wavelengths. Although optical filters limit the range of frequencies measured by a given detector, when two or more fluorophores are used in an experiment, there is often an overlap between the wavelength ranges. Compensation is the mathematical method used to correct for the overlap of one fluorophore's emission into another fluorophore's emission measurement. In order to determine how much compensation should be applied to the dataset, some control readings must be performed first. Analyzing data is a multi-step process that has been considered as the most critical parameter for biological experiments. In this presentation, I will only talk about the features of the Attune software to analyze data. The Attune cytometric software displays data in histogram, dots, or density plots. The histogram is a graphical representation of single parameter data and shows the relative number and distribution of events. In a histogram, the horizontal axis corresponds to the signal intensity of the selected parameter, while the vertical axis represents the number of events, the count, per channel number. A dot plot is a graphical representation of two parameter data where each axis represents the signal intensity of one parameter. One parameter is displayed on the x-axis and the other parameter is displayed on the y-axis. A density plot is a graphical representation of two parameter data where the colors represent the number of events with the same intensity and each axis represents the signal intensity of one parameter. Conventional flow cytometry analysis consists of drawing a region around a population of cells, which is called gating, and applying that region to other parameters within the experiment. This allows specific groups of cells to be selected for further analysis of other markers. Gates can be customized by using Boolean logic to link multiple gates together. However, the most popular application is to use gates in sequential order to limit data display and define the parameters for statistical analysis. Flow cytometry is widely used in various fields, such as immunology, molecular biology, cancer biology, microbiology, and so on. More specifically, flow cytometry is used, for example, in phenotypic characterization of cells, measurement of apoptosis markers, cell cycle analysis, and cell proliferation assays, quantification of transfection efficiency, and detection of single or multiple microbes in various types of samples. At the BioLab, we have the Attune Acoustic Focusing Cytometer by Applied Biosystems. It is a two-laser, red and blue, 6-color system. This device uses ultrasonic waves in combination with hydrodynamic forces to position cells into a single focused line along the central axis of a capillary, allowing the collection of more photons for high-precision analysis at superior volumetric sample throughput. This highly sensitive system is designed to acquire data at up to 35,000 events per second, detect particles with diameters from 0.5 to 50 micrometers, and rare events at sample rates up to 1,000 microliters per minute. The Attune software is designed to provide powerful data acquisition and analysis and can perform pre- and post-compensation automated or manual. As previously mentioned, the phenotyping of cell populations is one of the applications of flow cytometry. This means the identification and characterization of a specific cell group in the mixed population, for example, blood cells. Specific cell surface membrane proteins, which are known as markers, can be detected by using antibodies. Therefore, immunophenotyping of the cells can be accomplished by staining of a single cell simultaneously with two or more antibodies. In this example, we can identify different cell populations from a blood sample, analyzing a forward scattered versus side scattered dot plot. The smaller and less complex population represented by lymphocytes is shown in the red gate in the lower part of the dot plot, while the granulocytes with greater internal complexity are located in the upper part of the graph, and the monocytes of intermediate size and complexity in the central part. Therefore, side-scattered and forward-scattered analysis allows for morphological discrimination and quantification between lymphocytes, monocytes, and granulocytes, but does not allow the differentiation of cellular subtypes such as CD4 or CD8 T cell subtypes. This kind of profiling to distinguish morphologically similar subtypes can be made by using fluorescent markers as antibodies, which will characterize the presence of specific targets that identify cells. Observing the right panel of the figure, we can identify CD4 T cells gated from the lymphocyte population as indicated in the left panel that were stained with a PE-labeled anti-CD4 antibody. Flow cytometry can also be used to measure cell proliferation. The most common protocol is to stain target cells with the fluorescent dye carboxyfluorescein succinimidyl ester (CFCE). This dye is covalently coupled to the cell and can be retained for a long period. Due to their high stability, the fluorophore can be observed in the daughter cells, however reducing in density to each cell division. In this way, it is possible to monitor the cell proliferation of CFCE-labeled cells. In the figure right panel, we can see CD4 positive non-stimulated T lymphocytes labeled with CFCE. The signal is a sharp peak in the histogram. Stimulating these T cells in the presence of CD3 and CD28 antibodies induces cell proliferation characterized by the appearance of 5 additional peaks that exhibit different intensities of CFCE as a function of cell division. Cell cycle analysis is one of the pioneering applications of flow cytometry. During the cell cycle, there are alterations in the genetic content, which can be observed by fluorescent dye tags, such as propidium iodide. Rapid and quantitative measurement of apoptotic cells can be detected by flow cytometry. There are many different flow cytometric methods for the assessment of apoptosis in cells, such as measuring plasma membrane changes, detection of active caspase-3, and DNA fragmentation. As you know, apoptosis is characterized by a variety of molecular and morphological changes. One of the earliest events in an apoptotic cascade is the alteration in the plasma membrane. The membrane phosphatidylserine is translocated from the inner to the outer leaflets without affecting the plasma membrane integrity, which leads to the culmination of apoptosis by the loss of plasma membrane integrity. Double labeling with propidium iodide (PE) and fluorescent anti-annexin 5 antibody is a method to assess membrane changes. In the picture, left panel, we can observe lymphocytes cultured under normal conditions. These live cells that preserve membrane integrity are not labeled neither by propidium iodide nor anti-annexin antibody. When inducing apoptosis with dexamethasone, as shown in the right panel, we observed that the cells were labeled with anti-annexin 5 antibody, suggesting phosphatidylserine exposure, characteristic of apoptosis. Cells in advanced process of death also become more permeable and can be stained by PE. Another molecular hallmark of apoptosis is the activation of caspases. Caspases are inactive enzymes and must become active during apoptosis. One of the effector caspases activated at the late apoptosis stage is caspase-3. Active caspase-3 initiates a series of events that kill the cell. The active caspase-3 can be detected and analyzed with flow cytometry. BCL2 is a human proto-oncogene located in the membranes of the endoplasmic reticulum, nuclear envelope, and in the outer membrane of mitochondria. Preclinical studies demonstrated that BCL2 is anti-apoptotic intracellular protein, and the decreased BCL2 level indicates progression of apoptosis. In order to detect BCL2 activity, cells are fixed and permeabilized. Then fluorescence conjugated BCL2 antibody is added into cells and analyzed by flow cytometry. Programmed cell death is accompanied by degradation of chromosomal DNA. Technically, the easiest characteristic to detect is a loss of DNA from permeabilized cells due to DNA fragmentation. DNA can be stained with propidium iodide and detected by flow cytometry. The advantage of this method is that it is very fast, inexpensive, and simple. Furthermore, it detects cumulative apoptosis and is applicable to most cell types. However, the method may not detect cells induced to apoptosis in the G2M phase. An alternative method to identify DNA fragmentation involves the identification of the nicks or strand breaks using the tunnel technique. In microbiology, flow cytometry permits the reliable and rapid detection of single or multiple microbes and can provide information about their distribution within cell populations. Flow cytometry may also lead to a faster means of viability counting of microorganisms, while at the same time enabling a better understanding of all bacterial cells within a given population. Flow cytometry is used, for example, in environmental biotechnology, microbial community fingerprinting, clinical microbiology, and water analysis. Now, I will talk in more detail and give examples of some flow cytometry applications. In this paper, the antiproliferative activity against human colorectal, hepatocellular carcinomas, and breast adenocarcinoma cell lines of a copper complex was determined, demonstrating high cytotoxicity. Flow cytometry was used to demonstrate the induction of apoptosis and the delay in the cancer cell cycle progression. In this other paper, flow cytometry was also used to evaluate apoptosis as well as autophagy in the study of the cytotoxic activity of a platinum complex towards ovarian carcinoma cells. This one is an application example of flow cytometry in environmental biology. The Attune Acoustic Focusing Cytometer is capable of differentiate two major groups of microbes that comprise photosynthetic picoplankton in environmental samples based on their intrinsic fluorescence. The aim of this study was to evaluate non-viral gene delivery by a series of decatonic Gemini surfactant phospholipid nanoparticles and to explore their mechanism of interaction with cellular membranes of murine epidermal keratinocytes. RFP gene expression and cell viability status were evaluated using flow cytometry. To finish this webinar, I would like to recommend these resources for further information in flow cytometry. Thank you for your attention, and if you have further questions, you can leave them in the comment section of this video or contact me using this email. For more information about the BioLab, this is our website. Thank you once again. Take care.